Another study where we looked at Gulf War illness, chronic fatigue syndrome. We perceive Gulf War illness, which is very similar to chronic fatigue, as a subgroup, if you would, of chronic fatigue. And NK, and normal. It's very poor NK cell function in both those groups. Now, if you split the NK cell group into the chronic fatigue patients into fairly normal NK cell function versus abnormal NK cell function, you find that the SF36, a functional scale, and this is the global SF36 score, is significantly different between these two groups. So again, immune dysfunction is correlating with the severity of illness in this patient population. A different objective marker of severity, this is the PASIT. You've heard that a couple times. The PASIT is a cognitive assessment that studies executive function, how well your higher levels of thinking are working, if you would. And again, the low NK and the normal NK split, and the more severely impaired NK cell function, people are having more impaired cognitive function. So shift for a moment to this natural killer cell. What is this thing about? We've been talking about it not working well for a lot of years. Dr. Fletcher and the whole team in her laboratory are doing fabulous work with this, trying to figure out what is wrong with this cell. And one of the things we've looked at using flow cytometry is the stuff inside the cell that does the killing. It's a nice way to try to get a sense of, is there a functional defect of this cell? And you need to look at the perforins and the granzymes, the things that are chucked over into the target cell to kill the target cell. And we look at whether or not there's enough of that. And so we have the following data. This is intracellular perforin in the Gulf War and the chronic fatigue patients, and the normal controls. So they have abnormally low perforin content in their cells. And if you split people into low function for natural killer cells and high in the NK cell, there's lower perforins in the less functional cells. This is very interesting, and again, that Mac back there is messing up the word slide. They have that little advertisement between Mac and Microsoft, I think. Anyway, if you look at cytotoxic T cells, this is very important. Anyone who's an immunologist says, yeah, who cares about NK cells? Because, hey, all those cytotoxic T cells, they're so much more important. They're so much more efficient at viral killing than an NK cell. An NK cell could almost be seen as a backup system to these cytotoxic T cells. But here we have the perforin content of the cytotoxic T cell, and irregardless of NK cell function, the perforin is poor in the cytotoxic T cell. So cytotoxic T cell functional studies are really hard to do. You have to do all this complicated stuff. This is probably the only study I know of in chronic fatigue that looked at a surrogate for cytotoxic T cell function, and it says it's not there. We also look at immune activation through various markers, and one of the ones we like the best, because it's got some interesting functions, is the CD26 marker. It's basically sending the cell out of the periphery and into the tissues where it belongs after it's activated, and it has a lot to do with the regulatory function of immune activation, T cell activation. And again, we see it very high in the chronic fatigue and the Gulf War groups. We take a very large group, 168 chronic fatigue and 179 controls, largest one I've seen so far, and we see a significant difference across the board on the amount of CD26 expression on these cells. Now, this is an important thing to see. There are more cells that are expressing this. There's more activation. But on a per cell basis, the ability of the cells to actually put that marker where it is on the receptor, which is a very important functional marker, is actually quite a bit lower than the controls. So there's more activation, but the functional ability of the cell to express that marker, the quantities of markers receptor you need on that cell, is diminished, and it's a very significant thing. Now, an inexpensive way to look at that, because this is all flow cytometry so far, is to look at these markers that then fall off the cell, and you can measure them as a soluble form of the marker. And you can see, as it is on the cell, as it is in the plasma, that there's less soluble CD26 circulating. And we think this is a very good biomarker for circling the group that is chronic fatigue syndrome. We tried to do an index score. It helps a little, but it's not as good as I think the soluble CD26 on its own. So that leads us to the next thing here, which is that the neuropeptide Y is another soluble mediator that's circulating, and it's a powerful mediator. It's made by this it's being made by the sympathetic nervous system in the synapse of the sympathetic peripheral fibers, and it's cleaved by CD26. So if CD26 is down, neuropeptide Y might be up. Neuropeptide Y is a very active substance. It has many functions across brain and immune system systems. And so we looked at this thinking maybe this might be a biomarker that we could use in other ways. And sure enough, we find neuropeptide Y is elevated as compared to controls in a very significant way. More importantly, and I put this one in for you, Peter White, I hope you're in the room, you'll love this. All you guys looking for biomarkers for symptoms love this. It correlates in the right direction for many things. Neuropeptide Y, the higher it is, the worse the function of the patient. It also correlates with a number of the neurocognitive and mood states in the way that you would predict. So it's lovely. It might be the very first decent marker that we might use for managing that's accessible for looking at people across time and against severity of illness with specific mood states. This one just happens to be cognitive against neuropeptide Y, going in the right direction there. So, to sum, because the red light is flashing and it's lunchtime, to sum, the immune changes that we see in chronic fatigue syndrome I think are absolutely what you should see in a chronic viral state. The cells that clear viruses that are latent, that are trying to reactivate, the very cells that prevent reactivation of latent viruses are the ones that are most dysfunctional. It's an important point to be made, okay? And the state of chronic immune activation is something that would trigger baggage DNA, Dr. Williams, I say this for you, baggage DNA, and allow the virus to express what it knows how to do, including any latent virus stuff that's in there. So the retrovirus talk yesterday, Dr. Williams' talk on EBV-DUPTA is very, very important to have a chronic immunologically activated state to achieve that, as well as any virus that is actually capable of replication on its own. So the immunologic state seems very reasonable to continue to hypothesize viruses might well be involved in the chronicity of this illness. We've looked at a lot of reasons why they are dysfunctional. I think we're very far along in understanding that. I would say when you're looking at antiviral trials, just like in any other chronic viral state, you have to say what is going to be left if you cleared the virus. Is the immune system going to be healthy enough to maintain that? And you might want to be considering both immunomodulator and antiviral combination trials as time passes by and hope to be able to drop a person back into an immune system ready to hold them, if you would, once you get viral loads back down. The natural killer cell is a good surrogate marker for the severity of illness, the function, so is the perforin content, so is the granzyme content. It's also important to recognize the cytotoxic T cell is equally affected. Finally, these biomarkers coming from immunology land might be very, very useful in clinical trials. We're hoping to see both CD26 and neuropeptide Y put into a number of different studies we have ongoing, a lovely natural history study we call the Good Day, Bad Day study to see what's fluctuating with time, and we hope to be able to answer that question for you in some future conference. So I'd like to thank all the people that pay for our work, including the CFS Association of America, who are in the room. Thank you very much. And the conference organizers, because this is one fabulous conference. Thank you very much.